Assalamualaikum. Today my topic is PCR. PCR stands for polymerase chain reaction. It is consists of three words. The first one is polymerase, which means an enzyme. Chain reaction is compound word that means an action occurred in exponential way or an action occurred in step by step. Polymerase chain reaction, it is an enzymatic process in which DNA segment is amplified about million to trillion copies. PCR एक ऐसा enzymatic process है जिसमें DNA segment को copies किया जाता है about thousands to billion. पीसीआर लर्निंग ऑब्जेक्टिव्स, हमारे सीखने के मकसद कौन से हैं? द फर्स्ट वन इज हिस्ट्री ऑफ पीसीआर, सेकंड इज इंट्रोडक्शन टू पीसीआर, द थर्ड वन इज स्टेप्स ऑफ पीसीआर प्रोसीजर एंड द फोर्थ इज एप्लीकेशन और यूजेस ऑफ पीसीआर. History of PCR. PCR revolution method invented by Kary Mullis in 1983. Mullis was a great chemist in 1993. Mullis was awarded with Nobel Prize in Chemistry for PCR technology in Inver Valley, California. And this is a picture of Caribou Mullis. He was born in 1944 and died in 2019. Introduction of PCR. PCR laboratory technique that amplify the segment of DNA to form its thousands to millions of copies in an efficient way. पीसीआर एक ऐसा रिवोल्यूशनरी मेथड है जिसके जरिए हम डीएनए के thousands to million copies बना सकते हैं. What materials are used in PCR reaction? The first one is DNA template, second is tech polymerase, third is nucleotides, and fourth is PCR buffer, and the fifth is PCR tube. The first is DNA template, which is known as sample of DNA or segment of. The next is tech polymerase, which is type of an enzyme that helps in polymerase chain reaction. And the nucleotides, these are the basic units of DNA. And the PCR buffer which is used in polymerase chain reaction to maintain its pH. And the last one is PCR tube, which is used to detect the sample. This is picture of DNA template or the DNA segment. This picture shows all the materials which are used in PCR reaction. The first one is DNA template, second is nucleotides, and third is tech polymerase, PCR buffer, thermocyclers, and PCR tube. The steps of PCR procedure. It is consists of three steps. The first one is denaturation, second is annealing, and third is extension process. This animation shows the process of denaturation. It is also known as separation process, in which double-stranded DNA is separated into two single-stranded DNAs when temperature is raised above 94 degrees centigrade. And this animation shows the process of annealing. It is also known as binding process, in which the primers are bind to the single-stranded DNA when the temperature is lowered about 55 degrees. And this animation shows the process of extension or elongation process when the floating substance or nucleotides are bind together to form new double DNA stranded DNA when the temperature is higher about 72 degrees centigrade. One PCR cycle. The first reaction denaturation, second is annealing, and third is extension. And after that, double stranded DNA is formed. Gel electrophoresis. The use of gel electrophoresis and PCR machine. It is used to visualize the DNA. Gel electrophoresis is a technique in which fragments of DNA are pulled through a gel matrix by an electric current and separates DNA fragments according to size. जेल इलेक्ट्रोफोरेसिस एक ऐसी टेक्निक है जिसकी मदद से हम डीएनए को विजुलाइज करते हैं and this picture shows the process of gel electrophoresis when the DNA is going to be visualized. Application or the uses of PCR. PCR is used in genotyping, cloning, mutation detection, forensics, and diagnostics. These are the main uses of PCR. Genotyping. Genotyping is a process of determining difference in the genetic makeup of an individual by examining individual's DNA sequence. जीनोटाइपिंग यानी पीसीआर की मदद से हम किसी भी इंडिविजुअल का जीनोटाइप को आइडेंटिफाई कर सकते हैं, डिटरमाइन कर सकते हैं. Cloning. It is a scientific technique that is used to make exact genetic copies of living things. Genes, cells, tissues, whole animals can all be cloned. Cloning एक ऐसा प्रोसेस होता है जिसमें हम किसी भी जीन, टिश्यू, सेल या होल इंडिविजुअल का एग्जैक्ट कॉपी बना सकते हैं और पीसीआर की मदद से हम किसी भी एनिमल का या जीन या सेल को exact copy बना सकते. Next is mutation detection. It is important in all areas of biology. Detection of unknown mutation can involve sequencing of kilobases of DNA. हम पीसीआर की मदद से किसी भी डीएनए या जेनेटिक मेकअप में म्यूटेशन डिटेक्ट कर सकते हैं. After that, the forensics. It is scientific technique used in connection with the detection of crime. हम फॉरेंसिक्स की मदद से किसी भी क्राइम को डिटेक्ट कर सकते हैं और पीसीआर की मदद से हम जीन्स या डीएनए की मदद से क्राइम को डिटेक्ट कर सकते हैं. And the last one is diagnostics. It is concerned with the diagnosis of characteristics of particular species, genus, illness or other problems. हम पीसीआर की मदद से किसी भी स्पीशीज, जीनस और इलनेस को डिटेक्ट और डायग्नोस कर सकते